Okay, so I'm going to pause that. Okay, we'll start in about 4 minutes. It's a little brighter. Okay, we got 3 more. We got 3 on right now, looks like. Starting in under a minute. Okay, 6 o'clock. Okay, so since we've had a lot of PCR in this course, and since a lot of people have had various types of PCR problems, I thought it'd be good to go ahead and, or Josiah thought it'd be good to go ahead and run through PCR troubleshooting. So PCR is sort of trouble prone anyway, so everybody has trouble with PCR from time to time, especially when you're first starting out. It's really easy to run into all kinds of different trouble. And the issue with PCR is there's sort of so many different knobs you can turn that can change things, that you can run into situations where PCR is working great one day and then it stops working and you're not sure which knob to turn. And so the issue becomes how do you start the process of troubleshooting and not just start cranking on the wrong knob this way and that way and spend a long time trying to do that. And believe me, I've done that. So The first thing, we'll start kind of broad strokes, and I'd like to kind of focus down on some of the specific issues that you guys have had. So please fill up the chat with issues and questions. And Dave already submitted a gel, so we'll start with him. Now, sort of the broad strokes is the obvious things. Make sure that you did everything right. So the really common place to make mistakes is like pipetting errors, math errors, forgetting a reagent. If you forget to add your forward primer, it's not going to work. If you forget to add your polymerase, it's not going to work. And you mostly don't run into that unless you're doing a lot of different PCR, but it's easy to do. And sometimes PCR is just temperamental, and sometimes you can run one and you run it out on the gel and nothing happens, and you can just do exactly the same thing, run it again, and it'll work sometimes. And that's sort of the PCR equivalent of just like turning it off, turning it back on. But sometimes it's because you forgot something, sometimes it's pipette problems, sometimes it just is that way. Now, when you're looking at repeated problems, now that's the thing that you really start to look for. A lot of times what I like to do is I like to run 2 or 3 reactions simultaneously that are identical. That way, if it just doesn't work, then I have 2 more that I tried at the same time that might have worked. I usually always do at least 2. So if you're doing PCR and you're repeatedly getting problems and nothing seems to be working, there's a lot of different ways it can not work. And so the simplest way that it doesn't work is just nothing at all happens. You run your gel and you have nothing at all, just blank rows of nothingness. And that's maybe the most frustrating, but first of all, check that you have the right primers. You didn't mix them up, especially if you have a lot of different primers. Make sure everything's labeled correctly and all that sort of stuff. So just sort of do the due diligence on making sure that it wasn't some small, simple mistake, because that gets you a lot of the time. The second thing is make sure that your reagents are good, but since everybody here has brand new reagents, dealing with things that are several years old isn't really an issue, but it is sort of a general thing. And then check your thermocycler. So there's a couple of things there. First thing is make sure that you programmed it correctly, and that can also be a source for small, simple mistakes. I've done it a bunch of times where something's supposed to extend for 40 seconds and I accidentally put in 4 seconds. So make sure you check your times and your temperatures and your step order. But once you've sort of done the due diligence on all of that stuff, then you can start looking at tweaking things. But you don't want to start tweaking things before you've tried it the standard way. And PCR needs tweaking a lot, and that just is what it is. Now, the first thing that you have to look at is you have to kind of diagnose what's going wrong so you know which thing to start tweaking. First thing, if you have no product, that tells you a couple of things. So from a more broad perspective, if you're not getting amplification, then you're either failing for your primers to bind, or failing for them to extend, or you're failing to get exponential amplification. So if you have nothing at all, then something in here is failing. Your template isn't good, your primers aren't good, your primers aren't in the reaction, your extension time is too low, your annealing temperature is too high, and the primers don't stick. Something like that is sort of what you see in those cases. You can also add, you can run into whole different situations where you have incorrect amplification, and that can be in a bunch of different ways. So the first one, it would be just to have a randomly different thing. So you have your gel, and then instead of having your 700 base pair product, you have one that's 2,000 bases or 200 bases, and that can be non-specific binding. That can be template problems. You can have contamination in your template that has something else that has a different product. You can have too low of an annealing temperature, in which case your primers can bind to the wrong place in the template, and that's especially true if you're using a lot of template, or if you're using template that is big and complex, like a genome. If you're using plasmid, it's a lot less of an issue because there's only so many options for binding. Now, if you're using something like human genome DNA, there's 3 billion base pairs. So there's a lot of different options for things to bind to. Now, if you start getting up to 20 bases or so, you should have plenty of specificity, but sometimes genomes repeat. And it's rare for the whole thing to repeat, but it does happen, and especially if your temperature is too low, because that means it doesn't have to be a perfect repeat. If your temperature is too low, it can be a few base pairs off, still stick, and still amplify. And so you can get those things. In those cases, the simplest thing is raise your annealing temperature. And generally, if I'm getting none of my specific product, the thing that I'm really looking for, then what I'll do is I'll raise the annealing temperature several degrees. Because what will end up happening a lot of time is you'll raise your annealing temperature several degrees, and you'll start getting both. So you'll have your sort of wrong product and your right product. And then you'll raise it a little more, and you'll get less of the right product and more of the, or less of the wrong product and more of the right product, until finally you just have the right product. So if you raise it just 1 degree at a time, that's a kind of a slow thing. Some PCR thermocyclers are designed to do temperature gradients. I don't know if anybody has one of those, but you can kind of do several different temperatures simultaneously, and that's super nice. Mine doesn't, so I have to do mine gradients across time. So I just run it, change the program, run it again, change the program, run it again, change the program, run it again, which is aggravating, but it works. And so that's one of the things, it's one of the most sensitive things that you can adjust. And in general terms, going down in temperature on the annealing temperature gives you more binding of your primers, but less specificity. And more temperature on the annealing step gives you more specificity, but less binding. So you have to look at what your problem is and go the right direction. Now, another issue that can happen is your annealing temperature can be wrong, or excuse me, your annealing temperature can be right, but the extension temperature can be, or extension time can be, say, too long, and then you can amplify all kinds of things. So make sure that while you're in your extension temperature, that you're not going way over the size, because you can start running into weird stuff. So all these are pretty tightly constrained in the protocol that everybody already has. One of the things that makes barcoding a little bit different is that some of these sequences do vary. So sometimes your primers will not have perfect match to the template. And that's okay, that's kind of expected with barcoding, because you're trying to identify something that you don't know. Usually if I'm increasing the annealing temperature, I'll usually go 3 degrees, 2 degrees. When I'm using especially long primers, like some of the ones with the M13 tags on them, like we have for some of the plant and fish genotyping, I like to go a little bit more, because there's just more possibilities to run into. Primer design is also a big thing, but I'm not going to go into that too much here, because the primers are pre-designed. You can easily design primers to give you all sorts of problems. But if you look at existing primers that are kind of proven, standardish primers, primers you pulled out of papers, those sorts of things, you usually don't have those issues. If you design your own, there's a lot of rules you have to look at, and primer design becomes sort of a big issue. You can create problems with your reaction by bad primer design. And sometimes you have no idea why your primer doesn't work, and you just try a different primer, scooched over a bit, and it works fine. And sometimes PCR just fails, and you never know why. But another issue is, one thing I should have said in the part where I was talking about no amplification, is contaminants. Especially if you're dealing with not super pure samples. So the purification method that we're doing for this class is really raw. It's designed so that you can do it without needing more equipment and a lot of complex cleanup. So normally you would need at least a centrifuge to do a really good job of cleaning up your DNA. With us doing a really raw extraction, there's going to be contaminants, and that can make PCR much more difficult sometimes. Usually it works pretty good. Sometimes it gives you issues. Obviously, we ran into some of that stuff with the plant stuff, had to change some of the reagents and the protocol a bit, and are getting better results now. And obviously, we all learned that if you don't use the right primers, it won't work either. So those sorts of things are issues. Now, the PCR problems that are back in the sort of too much amplification range, you can also get things like primer dimers, which is where the primers, instead of binding to the template and amplifying, will bind to each other and amplify and make this little short product. And that little short product will just amplify and amplify and amplify and amplify, and it'll become this perfect template that's super short and runaway reaction and chew up all your primers. And what you'll see then is you'll see a gel with this little blob on the bottom, and that can be, you know, a bright band that's very, very small. Primer dimers can be an issue, and there's ways to deal with that in how you set up your protocol, how you design your primers. One of the issues with primer dimers can be actually in the handling of your DNA. And so if you have primers that are subject to dimerizing, you can, you know, for example, Taq polymerase can extend at room temperature, just more slowly. So if your primers are stuck together, even if it's only by a base or two, then they'll stick together at room temperature and just sit there. And Taq can come along while it's sitting on the bench while you're warming up the thermocycler and doing stuff, and slowly extend it, and you can start making primer dimers there. And then you get a much better template, and the next time it's more and more, and it can be stable and exponential in the PCR cycle. So one of the things you can do to deal with primer dimers is you can start your reaction on ice. And so you can I usually just crush up ice, put your PCR tube in the ice so that you keep everything nice and cold as you're loading it, and then you take your cold reaction that's too cold for Taq to work efficiently, and then you warm up your PCR machine. It comes all the way up to the first sort of hot step, and you put it straight from the ice into that. And that can work a lot of the times, especially if you are getting the good product, but you're also getting some that can usually work to sort of really make things crisp. And then there's also hot start. Hot start is where you can start your PCR cycle, put all but one, like the primers and the template and the water, in your tube, but not the Taq, not the polymerase. Load it into the thermocycler, let it come all the way up to 98 degrees or 95, whatever your protocol is running. And then from that temperature, when everything's nice and hot, then you add the Taq. Make sure you mix it up well, and then run it that way. And I've seen that help things. And that can help because there's no way for it to extend, and by the time the Taq is added, the primers have already separated. And that's, again, if you're seeing primer dimers, or sometimes maybe if you're seeing things like non-specific binding. So if you're looking at where you have multiple bands of non-specific product, that can happen too. You can get, when you run your PCR product, you can have several bands that are all wrong, and you can have your right band in there amongst them, or maybe there'll be a bunch of different ones, and that usually means the annealing temperature needs to go up. So again, you're trying to crank that dial from less binding up to more specific. So by raising the annealing temperature, fewer of your primers will be able to bind the template. And so, especially like I was saying with like a genomic template, you've got primers that are kind of half stuck all over the place, and you may want to combine that with something like a hot start or prepping on ice, because you may be getting some amount of extension that's running into making these little small products. So let's see, what else have we got? You can also get smears. Now smears is kind of like that, where you have a bunch of different things, but it's also but it's just a big smoosh. And sometimes that's a product of your electrophoresis. So sometimes you got something degrading your DNA, and you got just like smudge all through your electrophoresis. Sometimes that can be degraded leftovers from your template if you just packed a ton of template in there. And so you got to watch that, and one way you can test that is run your template. Just run the same amount of template that you're going to use in your PCR reaction. Like if you're using 10 microliters, run 10 microliters and then run your PCR reaction. And then compare, but dump the whole reaction in your well. And then look at your PCR product and see if you see the same kind of bands and smears. If your template is really degraded, like say like this is one I did from fish, and this was yesterday. I'm not sure I would trust it today, just because the NaOH is pretty aggressive, and even though I've added the tris to it, it's still a pretty harsh protocol, so it might be damaged and broken down. Once you extract your template from that reaction and put it in your PCR reaction, it's pretty stable. I mean, it's very diluted, right? So the NaOH is not a really big issue. But if your PCR reaction is, but if you're using template that you made a week ago, here pipetting breaks long DNA molecules like genomic DNA. It can, for sure, especially aggressive pipetting. But the issue is you're not doing a long segment of DNA, right? So you're amplifying like this little 300 or 400 base pair piece. So the probability that the break that you will see is between the two primers that you have is very low, right? It would probably be all over the place, but not necessarily right there. So if you were amplifying big segments, it would be an issue, or if you were doing something like, big sequencing. But if you're doing things like plasmid DNA, it's not really an issue. Plasmid is very stable. Let's see. So we talked about smearing, primer dimers, no amplification. So what kind of issues are most people seeing? Are we seeing more, like what are you actually getting on the gel? We'll start with David's while people answer that. So smear on gel before. Okay, so I was looking at yours. Let's do a screen share here. Okay, so alright, so we're looking at this right here. Now I don't know if I zoom this, if y'all will be able to see the zoom or not, but anyway. So if you look right here, you see these big regions. So this is about 1,500 base pairs. Sorry, Siri thought I was talking to her. Alright, so this band is 1,500 base pairs, this band is 500 base pairs. Now, this is your before PCR, so this is just running out your thing here, and if we look at your notes in the class, you put blah blah blah. Okay, well I remember. So from your notes in the class, this one was the NaOH, or no, this was the non-NaOH prep, and this was the NaOH prep. Is that right? That's okay. So this prep here is, and this is with and without RNase treatment. Now, from what you can see, it looks like the RNAs made no difference at all. I'm not sure exactly what protocol or how much or how long you let it work, but it doesn't look like the RNAs did anything, because this to me looks like RNA. It's a few thousand base pairs, probably about 4,000 or 5,000 base pairs, and it's red. So a lot of the dyes will bind orange to RNA and greenish blue to DNA. So this to me looks like probably mRNA, just raw out of the sample. This obviously is DNA. Comment in the classroom. Let's see. Nope, not that one. There we go. Okay. So this prep, I'm not sure exactly what protocol that is. Swabbed with RNAs, swab without RNAs. Okay, so 1 and 4 are no RNAs, 2 and 5 have the RNAs. So yeah, no real difference between 1 and 4 and 2 and 5, but there is a difference between the two preps. So let's see, NaOH prep. So the NaOH prep seems to have gotten more RNAs. I don't know what the protocol is for the other prep, but this one definitely seems to have cracked, or more RNA. This one definitely seems to have opened the cells a little bit better, though sometimes lots of stuff isn't great. So let's see, and then post PCR, so that prep with RNAs, that prep with RNAs. So did you add RNAs after PCR? That'll pop up in a minute. There's a pretty good delay between me and the stream. But just put in the thing whether or not you added RNAs before or after, or if that's just the template you used. So from here you can see a few things. For one, there's something very big in the well here. Sometimes this can be protein, sometimes this can be. I just swabbed vigorously with the NaOH. Okay. Yeah, that's what I usually do. Is I just, when I take the swab, I just rub it all around my cheek, put it in the NaOH, and then just swirl. I just spin the Q-tip around and around until it gets kind of cloudy, and then I kind of squeeze the liquid back into the thing, but not with my finger. I just press it against the side of the tube and throw it away, and then add the tris and go on. I've always had really good luck with that prep method with cheek swabs. Now you can see a few things. So you got some big stuff here. This is in your PCR. I have seen some preps do this, but this looks like maybe a little bit of sort of large genomic DNA still in your stuff, or who knows. Now this stuff down here, it's like 100 base pairs. This looks like non-specific or primer dimers down here. But you see right around here, so you got like 1, 2, 300 base pairs, and then this is the DRD4 protocol. So there's 2 different versions of that gene. Well, there's actually many different versions, but there's a range of sizes for this PCR product. So one of them is between 300 and 400 base pairs. That's sort of the normal one. And then there's the risk-taking behavior associated version that has a larger beads and stuff that bind beads that bind stuff that isn't DNA. Oh, okay. So this is a bead protocol. All right, so the it looks like this is your product. It's hard to say for sure with the sort of non-specific stuff, because it's possible it's just more non-specific stuff. You'd have to sequence it to know for sure. But it looks like this is your smaller product, but it looks like you may have another one up here, because you can see it on both. Now, this could also be more non-specific stuff, but it kind of looks to me, at least one possibility, is that you have both versions of the gene. And that's always possible with genotyping of nuclear genes. And that's the thing to bear in mind. The gene that we're looking at in this particular experiment, right, the DRD4 protocol, is a gene found on your nuclear DNA, right? So it's found in one of your chromosomes, and that, or one of your nuclear chromosomes. So that gene you have 2 copies of, because we're a diploid species. And that's something to bear in mind when you're looking at plants. Sometimes plants are polyploid, right? Yeah. So if you've got this version from you could have this version from one parent and this version from another parent. So you could have both versions of the gene and amplify both versions of the gene, because you have 2 copies of every gene. Now, if you're doing mitochondrial DNA, like later on in the fish one, where you're genotyping fish, that one targets a mitochondrial gene, and you're only going to have one version of that gene, because you only get your mitochondrial DNA from your mother. Where in this case, you can get one version from your mother and one version from your father. But you have a let's see. you have... okay, so one thing that might help is trying to clean up the template, because this is a little ugly, or using maybe less template. If you're getting all of this binding, then you can either raise the annealing temperature, or you can 1 microliter of prep for PCR, so it's all amplification. Okay, so yeah, if you're doing, but you can amplify all kinds of stuff. So remember, PCR can be really, really sensitive. So what I would do is I would run this reaction again, and I would run it at a higher annealing temperature. Whatever temperature you tried, I would raise it by 3, and then I would run it again and see what it looks like on the gel. Hopefully you get down to just 1 of these bands or just 2 of these bands. But even if you see 2, that doesn't necessarily mean that it's a problem. The other thing you might do is dilute your template a little bit. Or maybe use less cells. So maybe swab a little less vigorously. If you have just a lot of cells, then you can get all kinds of stuff. The other thing to bear in mind is you are also getting not just human DNA, so there's all sorts of weird things that you could conceivably run into with the DNA you can collect in your mouth. So keep the template, maybe lower the template, maybe raise the annealing temperature, but otherwise you got a lot of product here. And as these go away, these should get brighter. And so even if you're kind of trending in the right direction, it should stop looking so sort of even and almost smeary, but you can still see sort of distinct-ish bands. They're just very close in size. Let's see. 1 microliter prep. So does anybody else have one? I know I saw a couple others in the class. I'm not sure if those people are here though. Let's see, we had this one. So this one's from earlier. This is Dave's as well. This was really big. This was, if that's 1500, I'm guessing this is plasmid DNA. Whole plasmid on the right, no PCR product. Okay. All this stuff's getting a little older. It depends. Most of the time when I prepare my PCR, I don't do it on ice, and most of the time that's fine. Sometimes it makes all the difference in the world. Every PCR reaction is sort of its own creature, and you have to figure out what it likes. And the thing is, there's so many sort of knobs and dials that you can turn, that there's multiple solutions, right? So one thing that you can look at, if for example you're not getting amplification, is you can change the temperatures, you can change the amount of template that you add, you can change the amount of primers that you add, you can change, use additives like DMSO. DMSO, a lot of people just sort of, so should that help? I mean, maybe, if you're depending on what you're getting, right? As far as adding polymerase last, but I usually add mine last, just because it's the most expensive part. So if I screw up, I throw away template and primer, not template, primer, and polymerase, especially if you're using the expensive ones, because like Q5 and those sort of specialty ones can get pretty expensive. If you're using, for example, but for what you're getting, I would try icing it, because you are getting like some extra, and I would try some of those things. But if you're getting no PCR, which I think a lot of people are getting no product, one of the things you can try, which I don't know if you guys have, is like adding additives like DMSO. and that can help primers to bind. So it has a similar effect to lowering the annealing temperature, even though you'd think it would go the other way because it's a solvent. But anyway, and a lot of people just sort of throw DMSO at PCR reactions. They'll add like 3%. Just anytime it doesn't work, they'll toss DMSO at it, just sort of as a magic, just to wave a dead chicken at it kind of thing. But you can end up with a lot of different solutions. So you can use additives, you can change time and temperature and the quantity of things, and if you're really doing sort of advanced stuff, you can change things like, how many actual nucleotides are in your reaction, and like the chemistry of the master mix. Typically, I don't mess with that, so there's enough dials to be turning already. DMSO, it's a chemical. It's pretty commonly used in a lot of lab work. It depends on what you're doing, but I think every decent lab should have some DMSO, but it's not necessary to get PCR to work. But it is sort of fancy, and it's not the only thing. A lot of people add all kinds of different stuff. But if you're doing like, and also sometimes things like changing the volume in the reaction. So instead of doing a 50, doing a 25 can make a difference, especially if you're amplifying sort of big products. Doing bigger reactions seems to work a little bit better sometimes. I thought we had some more people who were having issues. Let's see. I dedicated a lot of time to the question and answer. Not getting many questions. Let's see. So, alright, let's look it over then. So let's talk about, until somebody gives me a question, I'm going to talk about primers. So this right here is one of the later reactions. Let's see if I can do this. Oh, nice. It's going to disappear as soon as I start working with it. But, so this, okay. So this right here is the fish one towards the end. And this is, I don't remember the name of the fish. Some species of fish is genomic DNA. And you can see that the primer doesn't perfectly bind. Now these have an M13 tail on the 5' end. And so that's just a sequencing site, so that the product will have those sequencing sites on each end. And it's the same up here. But you can see that the sequence doesn't perfectly match between the primer and the template. And in the cases of genotyping, that can happen. Now these genes that are picked for DNA barcoding are very conserved, so they don't vary very much, and they're always there. This one's a mitochondrial gene that you're not going to live if you don't have, at least if you're anything with mitochondria. And so even though there are small variations, they're long and they're very similar. So sometimes when you're doing things where there is variation genetically, you can get differences that you don't necessarily expect. So if you know the species and you know that this is a perfect match, then you can sort of theoretically calculate the binding temperature. But if there are variations, then, for example, these two aren't bound, so that's going to affect the amount of energy that is required to get this primer to come loose. So it's going to be less efficient at binding. And this over here. So if you were a species that had 4 or 5 places where it wasn't binding, or down here, if you had 4 or 5 places where it wasn't binding, then you could have a different annealing temperature. So especially when you're doing barcoding, you have to kind of play with it, especially if you're using an unknown sample, because these primers don't always bind the same to every species. And the product isn't always the same length. So if there are variations, then you can have changes in your extension time as well. And so, especially with barcoding, until you kind of have that reaction down, you have to get a sense of what's going on with that one. And sometimes you just have to start with the standard protocol and wiggle around a bit until you sort of bump into something that seems to be working, and then kind of go more that direction. Increasing the temp is the first thing. That's what I would do. What temperature did you run it at? I assume the one in the protocol. And that's another thing. So one issue I ran into, my first PCR machine was a used machine, and all my PCR reactions were garbage for a long time, and I finally realized that it was slightly out of calibration, but it was like 12 years old. So it hadn't been calibrated in a long time. And if your machine is off by a couple of degrees, especially during the annealing temperature, that can make a difference. So what I had to do was measure the temperature of my thermocycler. So as I was running it, I just measured it in each step, and I found out that it's a couple of degrees low all the time. So I just changed all my programs a couple of degrees high to compensate, and it all started working. So that's something to watch out for too, the calibration on your machine. But let's see. So this one, the fish one, makes a product about 700 bases. This one's a little bit different than the others that we do, because this one uses 4 primers instead of 2 primers. And that can add some extra complexity, but the primers are basically just different versions of each other. They're all very similar, and they all bind kind of in the same place. And if we model this PCR reaction, it goes PCR. And so here's this gene. It'll go from primer 1 to 2, and you can see none of these 5' tails bind, but it doesn't matter because we extend from the 3' ends. We'll model the PCR, and then we have these. And if you look, you can go detect common features. So it detected the M13 sites, and bam, there's your M13 site added because it was part of the primer. And now when you go to sequence it, you can just tell them to sequence M13 forward, and they'll sequence from here on out. And that's why they have those tails. Let's see. How did you measure it? I used a temperature gun. So you probably can use a lot of different things, but I use this. I love this thing. So it gives you a temperature readout. Now, but you can measure it, I'm sure, with any decent thermometer. You know, you might want to, if you are using like a glass thermometer or something, you're going to need to give it some time. So you'd stick it in there. I would add probably some mineral oil into the well itself, and that way it gets really good contact. Can we get the snap? Oh, sorry guys. Snap gene files. Yeah, I can send you this snap gene file. Now with the barcoding ones, those are you know, those go to all kinds of different things, so it depends on the species. So you'll have a variation depending on what species, but I can give you this one, and I can give you the file for this specific species, and I'll have to check which species it is, but I'll blast it. Now, if you're using like a glass thermometer, what I would do is I would just make a sort of calibration run almost. So I would program it to 95 and then put my thermometer and give it like 10 minutes or something, because the temperature variations, if it's not a really instant sort of temperature reading device like a temperature gun, if it's a glass thermometer or something, you're going to need to give it some time so that it can measure and change accurately, because otherwise it's just going to be you know, it's going to be there for 15 seconds if you try to use your PCR protocol, is what I'm saying, and that's not going to be long enough for it to show up. Let's see. And sometimes some thermocyclers have issues with hot spots, so like especially the older ones, sometimes the middle will be a little bit different temperature than the edge, so I always try to use the very center of mine, and I kind of build everything around those very center wells. Let's see. any other questions? I was hoping we'd have more PCR problems. But yeah. So these are the main ones. Non-specific product and no product, and both can be equally frustrating. Non-specific product, again, raise the temperature, maybe do start prepping everything on ice and try to prep things relatively quickly. You don't have to rush, but you know, especially if you do it on ice, you don't have to rush at all. And then make sure that you've double checked all your temperatures and times. Make sure that you double check all your other things. And you can also do a negative control run. So one thing you can do is you can run PCR without a template at all. Just use some water or nothing, or just fill it out to the same volume just with blank water, and then run everything again. So that will tell you if you've gotten anything in your primers, because one of the big problems that you can run into if you make a pipetting mistake is you can touch your template, or touch your template with the PCR with the pipette tip, and then pull up some template, put it in a reaction, and then go right into your primer tube with the same pipette tip. And now from then on, you've contaminated those primers, and those primers now have DNA in them from the template on that run. And then any subsequent reactions that you do with those primers will have that DNA added as well. So make sure that you haven't accidentally contaminated either your polymerase or your primers, and make sure that they're good. And the easiest way to do that is do a negative control, so run it without template. And then if it amplifies something, that tells you that you can also drop out other elements, like you can run it without a primer and try to narrow down where your contamination is, because it may just be in one element. Hopefully it's not in your polymerase. But if you are having a bunch of non-specific amplification, that can be it. Or you can have other primers get into your polymerase if you're doing multiple different experiments simultaneously, and you get some primer from one experiment in your template. You can get all sorts of weird stuff happening. So that's what I would do. For you, Dave, I would raise the temperature, I would do a negative control, I would prep it on ice, and go directly from the ice to a hot thermocycler. But I think that's about it. Let's do any last second questions. I'll give it a couple more minutes, otherwise we'll probably end early if we don't have any more questions. Okay. So, okay. All right. Well, next week hopefully we'll have some nice, pretty gels in the class file, and it will be interesting to find out if you've got two different versions of that gene, or if you really do just have one or the other. So. Alright, well, good luck with that, and I will end this. Hold on, should we know anything we should know for the plant prep and PCR? The only thing is just look at the new protocol. It's been updated, so there's a new DNA extraction buffer that's, this one has some detergents added to it, and then mortar and pestle, grind up the leaf, so on, and follow the new protocol, which solves some of the problems with the old one, and give that one a shot. If you have any PCR issues or gels from the plant PCR, do it. If I were you, I would, instead of just trying to focus down on the first one, I would sort of run all of these. So run each one and then run a gel on all of them. That way you can kind of save a little time and you don't get so far behind. Hopefully the plant one will work better than the DRD4 for you. Okay. I will see you guys next week then.